So hi everyone, I'm Lilly. I'm based here at the University of Birmingham in Alan McNally's group. And I'm here today to tell you a little bit about some of the research I've been doing in my PhD, where I've identified the loss of a type 6 secretion system in the successful MDR E. coli clone of SC131. And the work I'm presenting today is available as a preprint as of yesterday, so please feel free to check that out. So I'm sure I don't need to convince you of this, but antimicrobial resistance in E. coli is a problem. So this is a figure taken from a report in The Lancet that was published last year. And so basically it's ranking pathogens here by the number of deaths they've caused that have been related to AMR. And as you can see, E. coli is number one. And then on top of AMR being a problem, we also have multi-drug resistance. And so MDR is even more of a problem. And obviously this is because it limits the antibiotics that we have available to treat the infections that are caused by these bacteria. And something interesting that we found about MDR E. coli is that they seem to be better at colonizing the intestinal tract. So MDR E. coli are often XPECs, which stands for extra-intestinal pathogenic E. coli. And so how this works is that they can live in your gut, but then when they move to other parts of your body, they can cause infections such as UTIs or sepsis. And so how this works is that they are able to displace the resident nice commensal E. coli that we have in our gut. And this isn't great because if we have these MDR E. coli living happily in our gut, there is more potential for them to move elsewhere and cause infections, which is great for the bacteria, but not so great for us. And so the question that I'm trying to answer is, why are some of these MDR clones so successful at colonizing? Because we know it's not the resistance themselves, as this colonization process is happening in the absence of a selective pressure of antibiotics. And so we've generated a few hypotheses in our group. So we think maybe these MDR clones have some kind of enhanced metabolic capability that gives them a competitive advantage in the gut compared to other E. coli. Or maybe these bugs have the ability to secrete some kind of proteinaceous substances or some weird metabolites that they are able to utilize better than other E. coli, again, giving them a competitive advantage in certain environments. And we also think that maybe it's because they have a type 6 secretion system. So you might be asking, well, what is a type 6 secretion system? And to keep it brief, they kind of look like this. So they sit between the inner and outer membrane. And essentially they're little harpoons that some bacteria have that they can use to shoot out things like toxins into neighbouring bacteria, so they can use it as a weapon, essentially, to kill bacteria that are close by that they are competing with. But it can also be used for nutrient uptake and manipulating host cells. And type 6 secretion systems in E. coli aren't particularly well studied, and they're definitely understudied in MDR E. coli clones. So this is something that I wanted to look at. And so to do this, I thought I was going to look at SC131. So SC131 is a lineage of E. coli that contains a really big MDR clone, and it's a really successful one. So this is a picture showing the emergence of SC131 in the clinical landscape in the UK. So you can see here it emerges in the early noughties, and it's kind of dominated and become one of the prevalent lineages that we see causing bacteremia in the UK. And this is a similar study that was done in Norway that shows a similar pattern. So you can see in grey here, we have the SC131, and you can see that it's really kind of dominating the scene over in Norway as well. And so we've got our MDR lineage that we wanted to examine. And so what I did to start with was, let's have a look and see if there is a type 6 secretion system present. And so to do that, I gathered a data set of just over 3,000 SC131 genomes. And I took a load of type 6 secretion system genes from an online database called Secret6 and just looked for them in this large genome set. And something that's really good about SC131 is that the structure of it. So there are three clades, clade A in the dark blue, clade B in the cyan, and clade C in the magenta. And you can see this really long branch here. This is the MDR clone. And what's nice about this is that the evolutionary history is quite clear. So clade A and clade B are much less drug resistant. And then clade C has evolved and gained a big MDR plasmid and a few other types of resistance through SNPs. And it's basically just a nice way of tracking and comparing like an ancestral susceptible lineage to the MDR clone that we're interested in. So going back to the search that I did over the 3,000 SC131 genomes, this is the result. So we have the presence marked in blue. So we have the type 6 secretion system genes across the top, and then the phylogeny on the right, on the right. So we have the clade A here, and clade B, and then the big MDR clone within clade C. And for the majority of genes, you can see there's not really any distinction between the susceptible and the drug-resistant clade, apart from this one gene here, labelled as VT4E. And so you're probably wondering, what is VT4E? Because that's exactly what I was thinking. And so I did a little bit of literature digging, and then I also did some blast searching, and I found that VT4E was 100% identical to a gene called VAS-K, which is found in Vibrio and is part of a type 6 secretion system, of course. And then we found that the homologue of this in E. coli is called TSSM. So basically we found that between the ancestral susceptible E. coli and the MDR clade C that we're interested in, the only real difference is the presence of this TSSM gene. So it appears that the TSSM gene is lost within the drug-resistant clade. So we know what's happened, but really we want to know why this has happened. Why has this MDR clone lost this TSSM gene? And so to do that, I wanted to start off with a good reference sequence of the entire type 6 locus in SC131, as that wasn't available. And so I found a nice clade A strain, and I sent it off to Microbes NG for sequencing. And then from that, we were able to characterize the full locus. So we have all the structural genes colored in purple. And you can see that type TSSM is a very big structural gene. And so now we have this really good reference. I could then take a clade C genome and compare it to this to see what was happening where TSSM should be. And what we found was that indeed TSSM was lost. And what had happened is that an insertion event had occurred. So at the top, we have the naive TSSM that was found in clades A and B. And then this is the case for what we found for the clade C genome that we'd looked at. So there'd been an insertion event where we had ISEC12. And what had happened is essentially it had hopped into the middle of TSSM. And so it had been interrupted, and therefore this gene wasn't functional. And we just wanted to double check that this was the case for every clade C, and it wasn't just something that had happened in the one genome that I was looking at. So what we did was we did a search for the naive TSSM gene, the complete one, and then we also took a little bit of this insertion event, so half of TSSM in the start of the IS, and then the right side of it as well, and we searched for these three sequences. And you can see that we've confirmed that this insertion event has occurred in the whole of clade C. So you can see here we have the naive in the clades A and B, and then we have either side of the insertion sequence pretty much present in the majority of clade C. So now we know what's happened and why it's happened. So now we want to know, okay, so is this having a phenotypic effect? So I designed a competition assay. So what we were doing is we would take a clade A genome and grow it up and then give it a prey. So that was the lab strain, MG1655, and we'd co-culture them and see what happened, see whether it was able to kill the prey. And then we did the same with clade C. So growing up the prey, so we have the growth of the prey in monoculture, and then you can see in the presence of the clade C genome, the growth of the prey is greatly reduced. So we can see that the clade C is able to have some kind of, is able to outcompete the prey essentially, without the presence of a type 6 secretion system. And then we did this with the clade A. So this is the genome that does have a full type 6. And interestingly, we see that the prey isn't really reduced in the presence of this. So, okay, so we have something going on. We have this ability of the clade C to outcompete the prey, but it's not because of a type 6. So we can kind of rule that out on our list of hypotheses. So then we wanted to look, okay, so how about the extracellular products hypothesis? And so to do this, we designed a very similar experiment where we had the prey, grew that up, And then we also grew up the clade C. But instead of co-culturing those, what I did was I basically span down the clade C and then just cultured the prey with the supernatant of it. So this would capture any metabolites that had been released by the clade C, or if there were any phages or things like that that could have been accounting for the results that I'd found. And so these are the results, and you can see that the same pattern of reduction in the growth of the prey wasn't found. So this kind of tells us that it's not something that the clade C is releasing that's allowing it to outcompete. It's something to do, well, we don't actually know what it is yet, but we know it's not because it's secreting proteinaceous substances, or there's no interference of lytic phage or anything like that. And so that leaves us with the hypothesis of enhanced metabolic capabilities. So this is our current line of investigation in the group, and so please look out for any papers that we have in the near future that will be addressing this in more detail. But to summarise the work that I've done, I've shown that the type 6 secretion system is lost within the MDR clade of ST131, and that was due to the insertion event occurring in the structural gene TSSM. And then I've also shown that ST131C also outcompetes without the presence of a type 6. And this is why we think maybe it's something to do with metabolic capabilities that we are still looking into. And so with that, I would just like to thank everyone that was involved in this work. Again, you can check it out at the preprint if you'd like. And I would also like to thank the whole McNally group for their support, and of course, MicrobeNG for their sequencing services and having me here today.